Before we move on to the electron transport chain, a TCA review question. Press pause. Resume when you are ready. This is classic MCAT format, i.e., an interruption in a metabolic pathway will cause accumulation of which substrate. Without FAD, we cannot produce fumarate. The production of F-fumarate yields F-FADH2. One step upstream of fumarate is succinylate, choice D. We finally made it, Jackson, the electron transport chain. The ATP jackpot is produced through oxidative phosphorylation. Oxidative because it requires oxygen. Remember, Gatsby? Without oxygen, only substrate-level phosphorylation can take place. There are two components of the ETC, electrons and protons. These orange electrons are passed from NADH and FADH2 down a series of inner mitochondrial membrane-associated electron carriers, inner mitomembrane. These gray protons are pumped out of the mitochondrial matrix into the intermembrane space. This electrochemical gradient will be harnessed by the ATP synthase. Note that complex 2 does not span the entire width of the inner mitomembrane and therefore does not participate in generating the proton motive force. Here we have a chart outlining the four complexes of the ETC. I would like to point out complex 2, succinate dehydrogenase. Does that sound familiar, Jackson? This is the exact same enzyme from the TCA cycle. In fact, succinate dehydrogenase is actually bound to the inner mitomembrane. Also, note ubiquinone, aka coenzyme Q, an inner mitochondrial membrane shuttle, bringing electrons from complexes 1 and 2 to complex 3. Cytochrome C is responsible for transferring electrons between complexes 3 and 4. Let's take a closer look at cytochrome C oxidase, the fourth and final complex of the ETC. Cytochrome C oxidase transfers electrons to molecular oxygen, the final electron acceptor of the ETC. This reaction generates water.
Aye, oxygen. Sweet, sweet oxygen. Thanks, lads. Come back soon with some more oxygen for me.
Protons then flow down their electrochemical gradient into the mitochondrial matrix through the ATP synthase. This proton flow causes rotary movement of the F1 subunit, and voila, adenosine diphosphate joins with inorganic phosphate to produce ATP. This process is known as chemiosmosis. And our ATP scoreboard is complete. The ETC utilizes the proton motive force to generate ATP. One NADH generates 2.5 ATP, and one FADH2 generates 1.5 ATP. Some sources may give higher exchange rates, a difference due to variation in theoretical ATP yield as compared to actual ATP yield. Press pause. Resume when you are ready. The correct answer is A, cytochrome C oxidase, the fourth and final complex of the ETC. Notice that the complexes are merely in reverse order, T, C, B, A. Before we leave the mitochondrion, I must say it. #endosymbiosis.
With all this studying, I think it's time for some exercise. Let's go pump some iron.
In order to understand skeletal muscle, we must first understand its macro and micro components. Let's begin macro and move micro. Jackson's bicep is an example of skeletal muscle. Skeletal muscle is striated muscle under voluntary control. A muscle fiber is the same as a muscle cell. This single multinucleated cell is composed of many myofibrils. Myofibrils are highly organized arrays of repeating sarcomeres. A sarcomere consists of alternating bands of actin and myosin, which give skeletal muscle its striated appearance. Actin filaments are thin, and myosin filaments are thick. Now let's move micro to macro. The sarcomere is the functional unit of muscle. A myofibril is a group of sarcomeres. Muscle fiber equals muscle cell. Glucose is to starch as a muscle fiber is to skeletal muscle. Monomers aggregate to form polymers. When Jackson wants to flex his bicep, neurons in the motor cortex of his frontal lobe relay a signal to his spinal cord, which further relays this signal to a motoneuron. And we have arrived at the NMJ, the neuromuscular junction, the interface between a motoneuron and a muscle fiber. Press pause. Resume when you are ready. The correct answer is E, 25431. Step 1. Depolarization of the motoneuron. Step 2. Calcium enters the motoneuron terminal. Step 3. Exocytosis of vesicles containing acetylcholine. Step 4. Acetylcholine binds to nicotinic acetylcholine receptors at the motor endplate of the muscle fiber. Step 5. Sodium enters and potassium exits the muscle fiber. The sodium current dominates, serving to depolarize the muscle fiber. How is an electrical stimulus converted into mechanical motion? The answer is excitation-contraction coupling. Who's the major player? Calcium. Muscle contraction is essentially a calcium party. Aside from actin and myosin, what are the other components of a muscle fiber? T, or transverse tubules, are deep invaginations of membrane contiguous with the plasma membrane. T tubules provide quick and coordinated depolarization. The sarcoplasmic reticulum is a modified form of smooth endoplasmic reticulum, which stores calcium through primary active transport. ATP is bound to the head of myosin when myosin is not bound to actin. Tropomyosin is a protein that hides the myosin binding site on actin. Troponin C is the site of calcium binding. C is for calcium. Step 1. Depolarization is transmitted to the transverse tubules. 2. This signal is transmitted to the sarcoplasmic reticulum. 3. Calcium channels of the SR open. 4. Calcium diffuses into the cytoplasm of the sarcomere. 5. Calcium binds troponin C. 6. Tropomyosin moves, exposing myosin binding sites on actin filaments. 7. Myosin binds to actin. At this time, myosin has ADP and phosphate attached to its head. 8. The cleavage of this phosphate group initiates the power stroke. During the power stroke, the myosin head changes position, causing the actin filaments to slide past one another. 9. ATP binds myosin, resulting in myosin release of actin. If the calcium party is still going on, the cycle continues. If not, muscle contraction ceases. Press pause. Resume when you are ready. The correct answer is A, bound to actin. Myosin that is not bound to actin is bound to ATP. ATP is needed to break the actin-myosin bond. However, without sufficient ATP, i.e., following death, after which aerobic respiration ceases, myosin is constitutively bound to actin, thus producing a rigid corpse. Rigor mortis, a term derived from Latin, rigor meaning stiffness and mortis meaning death. Eventually, enzymatic decomposition ensues and the corpse will no longer be rigid. Myosin filaments are anchored at the M line, M for myosin. Actin filaments are anchored at the Z line. The sarcomere unit is measured Z line to Z line. The A band corresponds to myosin and includes the H band. The H band is exclusively myosin, whereas the I band is exclusively actin. I remember that the letter H is thicker than the letter I, as myosin is thicker than actin. Press pause. Resume when you are ready. The correct answer is D. Both the H and I bands shorten. Now here's a mnemonic you won't forget, Jackson. His shrinkage. The H and I bands shrink as the Z lines move closer together. The thick filament, or A band, is always the same length.
I am flabbergasted. Run that by me one more again. Contract. Relax. Contract. Relax.
Thank you, Prezi, Wikimedia Commons, Gatsby, Keto the Kitten, DJ Ilium, and of course, Jackson.